Okay, so then we move from stomach. Next in line in terms of secretion is going to be the pancreas, right? The pancreas sits, well, you've seen it now, so you know exactly where it sits. The pancreas sort of looks like a tadpole, right? Head and tail, right? And it sits in the little crook between as the stomach becomes the duodenum and the duodenum makes a C shape, the pancreas sort of lives in that little crevice. The pancreas is part of that very complex duct system, the biliary ductal system, right? So the liver is right here, the common bile duct comes down, and of course enters the duodenum at the sphincter of Oddi. Did you call it the sphincter of Oddi or the papilla of Voltaire? The papilla of Voltaire. So the papilla of Voltaire is the head. The sphincter of Oddi is the muscle that controls the gap, right? So right here. Now that little tiny place in the GI tract is incredibly important because that's where the bile duct and the pancreatic duct both dump their secretions into the GI tract, right? So it's a weak spot in our anatomy. If you were going to redesign the human being, you'd give the liver its own papilla and the pancreas its own papilla, right? But we don't get to do that. Okay, so big compound gland. Pancreas does two main things: digestive enzymes, bicarbonate solution. Okay, the bicarbonate solution is to neutralize the stomach acid. The digestive enzymes, the pancreas is the only organ in the body that produces enzymes that digest everything we eat, right? So proteins, carbohydrates, and fats, the pancreas has, and even things like nucleotides, the pancreas has an enzyme for that, right? So it is our primary source of digestive enzymes. Running down through the center of it is the pancreatic duct, and the little pancreatic acini dump their material into that. So the enzymes are produced in the acini, that's the little tree-like parts, right? And then the pancreatic duct is where most of the bicarbonate production is happening. All right, so here's our enzyme list. Make a little asterisk by this because I do want you to know these. Sometimes we have to replace these in patients, so they're worthwhile to know. Trypsin, chymotrypsin, and carboxypolypeptidase, those are all protein digesters. Now, they're going to digest proteins in slightly different ways, but they still are all protein digesters. We'll talk more about how they work after the test, okay, when we get to the metabolism discussion. So these are all the protein digesters. Common theme is the pancreas produces enzymes and releases them in an inactive form. Hugely, hugely important, because if they were in active form, the pancreas would eat itself, right? That's autodigestion, and that does happen. Okay, one of there's a disorder called pancreatitis. One of the forms of pancreatitis is where the pancreas is basically eating itself, right? So what's supposed to happen is they're released in inactive form, and then they're not activated until they get out here into the intestine, where the material they're going to work on is. But what happens in some forms of pancreatitis is the duct is blocked, right? So then these enzymes end up self-activating because of inflammation, and then you get this big cyst where you used to have a pancreas, right? Now you can live without a pancreas, but it's tricky, right? Because you're gonna need to be on insulin, and you're gonna need to take a boatload of digestive enzymes to get any nutrition out of the food that you eat. Okay, so let's see, I think we got all that. Pancreatic secretion. The primary stimulator of bicarbonate secretion is secretin, okay? Cholecystokinin is the primary stimulator of enzyme formation of the pancreas. And we'll talk a little bit more about that later on. All right, so last bullet point, this slide is protein digesters. Now let's look at the other digesters. Pancreas also produces a pancreatic lipase. That's our primary fat digester. Our pancreatic lipase is how we break down most fats. There's a cholesterol esterase, which helps us to break down cholesterol, and there's a phospholipase that helps us break down phospholipids. And by now, you've seen all of those. Cholesterol we find in cell membranes, phospholipids we find in lots of places, including cell membranes. And there's fats, of course, in many materials that we eat. So that's a lipid digester. Pancreas also produces a trypsin inhibitor, okay? And what the trypsin inhibitor does is basically it keeps trypsinogen, which is one of the protein digesters, intact until it's released, right? So it helps to inhibit self-digestion. Now the problem, again, the trypsin inhibitor system gets messed up in the case of inflammation. So like if the duct is blocked, then we'll end up with a lack of inhibition and autodigestion that happens. All right, so if the duct of the pancreas is producing bicarbonate, well, we like to know how that's happening. Okay, so here's our ductal cell. Here's the lumen of the long pancreatic duct. Here's the blood, right? So interstitial fluid. Okay, so outputs, we've got a bunch of bicarbonate secretion. All right. Now, where did the bicarbonate come from? Not surprisingly, it came from CO2 and water, right? CO2 and water combined, we get bicarbonate and H+. In the pancreas, the H+, here's our sodium hydrogen exchanger, right? And you'll notice that it's arranged opposite the gastric one, right? So here, the hydrogen ion is going into the blood, and the bicarbonate is going into the lumen. So it's going in opposite direction. Okay. So this is, this is probably going down its concentration gradient, I suspect. And the sodium is also going down its concentration gradient, which is why we don't see an ATP there, right? So this is a co-transport. All right, so we have our bicarbonate being released from our usual sources of CO2 and water. All right, how much is this? So the solution ends up being, has quite a high concentration of bicarbonate. So 5 times the concentration of the plasma. So basically we have a built-in acid neutralizing system, right? Bicarbonate's made, neutralizes the stomach acid. So as soon as the stomach empties, the bicarbonate solution is already right there in the duodenum, right? So it neutralizes the stomach acid before the materials continue. Let's see. I think we talked about all those things already. Oh, this last bit. As we concentrate the bicarbonate in the solution, we end up making this secretion hypertonic, right? So that's because I just said we concentrated this 5-fold, which means that there's a tendency to pull water and sodium in by osmosis. Okay, that's what this last line is here, is then that last bullet point. It just moves by